My name is Vogan, and I'm an editor with Nature Genetics based in Boston, Massachusetts. It's a real privilege to be able to participate in this meeting. Thank the organizers for having me. And it's my privilege to be able to moderate this first panel discussion. So the title of the session is Affordability and Feasibility of Large-Scale Genetic Risk Profiling. We have three panelists. The format is we have a series of questions prepared that I will pose to the panel, give each of the panelists an opportunity to respond to the question, and then we'll open it up to the audience to follow up with questions to the panelists or add their own thoughts on the questions that are being posed. To again introduce our panelists, so we have José Bras, who's filling in for Kari Stephenson. He's at University College London. Done a lot of work on neurodegenerative diseases such as Parkinson's disease, Alzheimer's disease. To his left, we have Jerry Schellenberg from the University of Pennsylvania. Again, made a lot of seminal discoveries on early onset Alzheimer's disease risk genes, and then, of course, has been an instrumental leader in several of the large-scale collaborative projects for Alzheimer's disease, genome-wide association, and sequencing-based studies. And then to his left, we have Cornelia van Duijn from the Erasmus University Medical Center in the Netherlands, doing a lot of, again, collaborative work, international projects on Alzheimer's disease and a lot of other common diseases. So I think, of course, they're well known to many of you in the room, so I think that introduction should be enough to get us started. So again, framing broadly the theme of the session, we can maybe restate the question as what is the current state of the science with respect to genetic risk profiling? Where are we now? Where do we need to go? Where are there still gaps? And then drilling down a little bit more from the standpoint of technology and study design, what are the best strategies that we can use to collect the data that we need in order to build accurate models for predicting disease risk? So with those broad questions to frame, we'll get into some specific questions. And the first question is on the theme of genome-wide association. So the question to the panelists is, should we continue to do ever larger genome-wide association studies, or have we reached a point of diminishing returns from the analysis of common variants?
Can I start? Well, I know a lot of teaching, and then I always have this nice slide saying predicting the end of GWAS is like predicting the end of the world. We all know it will happen once, but it's definitely very difficult to predict when the world is going to end. And that is definitely still the status, right? If you think about what we're finding in GWAS research, in Alzheimer's we haven't done the GWAS without finding new genes. And that's a fact, so we're finding new genes. The point is, if you look at it from a biological perspective, you can't help noticing that we're getting back and over again to the same pathways. So we're not finding a lot of new pathways. And that's a fact, I think, that we have to start working with now, not only from the point of biology, but also towards translation. It seems that if we look at the pathways, the lipid, the innate immune system, or the immune system in general, and then the endocytosis as a pathway, and the amyloid and tau pathway, that seems to be the major key drivers of the disease. So there is still an incentive to do larger GWAS, I think, because the question that we have to start answering is, how do the different genes and the pathways relate to the clinical spectrum, for instance? We're starting only to do that now, linking gene families and pathways to certain phenotypes that are very prominent in the disease. So, for instance, clinically, if you look at the disease, you see definitely in the early onset amyloid is the driver of the disease, right? We work a lot in Rotterdam with the elderly, 80 plus, 90 plus, and these people come up with cardiovascular disease and diabetes as major pathways that you see occurring in them, and there is some interaction there. And it's only now, and that's work that we did in the Paris Consortium with Julie, that we start seeing that these are perhaps immune-related phenotypes that are popping up in the elderly. So I think there is an incentive to do more work, both towards understanding the clinical heterogeneity of the patients, but also understanding the gene-environmental interactions, which are bound to be happening not on a gene basis, but on a pathway basis. And if we want to do pathway interactions on a genetic level, we probably will have to know the genes that are the key players and then build a kind of risk score based on that and look at interaction on that level. So I'm afraid that more data are needed. It will be wonderful to have more data and understand the disease better. But then not so much with the view for biology, but just translation. Let's go there.
So I thank Cora for organizing this really intriguing day. I would agree with Cornelia. I never disagree with Cornelia. It's just no percentage in that. But I do think we need larger GWAS studies. As you know, I always look at cardiovascular disease genetics as a model, and we're nowhere close to the hundreds of thousands of cases that they have used in their studies, and those studies have revealed very interesting genes, including some which are therapeutic targets. In terms of, you know, we have very large Caucasian GWAS populations. We don't have large, for U.S. African American or Hispanic populations or Caribbean Hispanic or Asian populations. And what we're able to do is we're able to look in those populations at the known genes, and when we do that, we see very different allele frequencies in different populations. So different genes have much greater or much less influence on a particular population. So, you know, our rationale is there's more, if we're looking at different ethnicities, there's more to be discovered, and there will be genes that have a larger effect size in a particular ethnicity that we will miss in Caucasians if we don't go into that other ethnicity, even though it may not be, they may be actually active in both populations, so we need to do that. And of course, we need to be able to treat African Americans in the U.S. Along with Caucasians, and that treatment may be different in terms of their most prominent genetic features. So I think increasing the Caucasian data set is important, and I think just for the common variants, which I think this question addresses, that's true, but also expanding our sample size is going to greatly help with rare variants. Then there are phenotypes that we do not have good populations for example, different PET imaging, tau and A-beta, and immune imaging compounds that we don't have enough people that we have those phenotypes on. CSF biomarkers, again, we have on the order of 4,000 to 5,000 people with tau A-beta CSF biomarkers, not 50,000 that we need, and that's going to tell us different things. And then one of my constant themes is structural variation, which accounts for half of the genetic variation in the genome compared to single nucleotide variants, and we don't, that's the dark matter of the genome. We do not have a good technology for measuring structural variants, and when we do, some of those will be rare, and we will need large populations that we can tap into, and some point we'll be able to impute structural variants, but we can't right now. So that's another reason for not only doing a bigger GWAS, the GWAS is the starting data, but just having that bigger population for all these other studies.
Well, it's great to be the third because I don't have much to say. I think I agree with everything that was said. I'll say, however, that I'm always a bit concerned, and this comes back to what Cornelia said. I'm always a bit concerned because there's a chance that we won't find anything else that's relevant in the next GWASs that we do in these diseases. It's what Cornelia said. We found all of these genes. We're still finding genes. They lead to the same pathways. And so I agree, we need more samples. This will give us more hits. These hits will likely be in the same pathways, different, players in these pathways, but maybe there won't be any new pathways to find, and if there are, maybe their effects are going to be so small that is there really a point in having them when we think of translational science? And I think Valentina's work has shown that what we have so far really captures almost everything that we can find in terms of polygenic risk for Alzheimer's disease. And so I'm all for doing more genetics, of course. What Jerry said, it's obvious we need to do much more in different populations. I mean, we've focused on the Caucasian population, and this is it's only a subset. There's so much that we can learn by doing other populations. I think this should be a focus. I think people in the US really are driving this. But I'm always worried that we're doing all of this and we'll find new genes and they'll just be on the same pathways and they won't be informative. So that's just my thoughts.
So to the audience for comments and questions.
First, I think that when there's quality, it's very one comment, and that is as the N goes up, the quality control inevitably gets more difficult to do. And, and that, I mean both the quality control in terms of clinical diagnoses and also the quality control in terms of ethnic background. As we stretch outside our comfort zones and we already know, for example, that the Alzheimer's centres have, misdiagnosis rates of, let's say, 15 to 20%. And I worry that as we increase our N, we will make more false findings, even those which exceed the Bonferroni correction. That's one thing that worries me. On the positive side, on the other side, one thing I think we have not done anything like enough, and I'm surprised Jose didn't mention this, was, is look at other phenotypes like DLB, for example, to play the ball into his own court area, and PSP, which is a tangle disease. For these, our GWAS are very small, and yet they could profitably tell us a lot about the pathogenesis of Alzheimer's disease. So I would plead for more GWAS, especially of rarer phenotypes, but a bit of caution about just increasing the N of, for example, the Alzheimer GWAS.
Can I comment on that? Because I think that what you worried a little bit of is the big data approach, right? And you must have seen the paper on in the archives on the UK Biobank that even a proxy assessment of diagnosis being a positive family history seems to work, right? And I'm afraid that if you say what will be the novelty, how we can move this field forward, I think that the answer will be in part in getting the data bigger, but using also big data analysis, right? And that means that you have a lot more noise in everything, in your diagnosis, in your genetic assessment. There's also more noise, whether you sequence or GWAS, there's noise. But what we have to start doing, I think, in genetics, but definitely also on my other side, I'm also an epidemiologist, is using also the big data analysis in combination then with the proper follow-up, being epidemiological translation into independent data sets. And I think that will be about the only way out. Although I think if you look at it, what we've done in statistics, in genetical statistics, we always have been able to solve the problem, right, so far. So I'm not so worried that there's a lot of noise that we cannot deal with statistically, because up until now, all the bright minds in the world, whether it's Wellcome Trust or Broad or anywhere in the world, there's always been found solutions for the problem. But I think part of the answer will be if you talk about phenotypes, like verbal kind of problems. A lot of patients with dementia present with verbal problems, right? These are all subgroups that we don't understand at all, and it's going to be important. Because we try to still attack the dementia and Alzheimer's disease as one disease, but it's not going to be one disease, just like myocardial infarction wasn't one disease, right? And come on, if there is different etiologies underlying myocardial infarction, you can have endocrine problem, your glucose metabolism, you can have your blood pressure up or down, or lipids disturbed. You have to tackle all of these specifically, and the guys and the women in cardiovascular disease can assess these in the blood. But the only way we're going to be able to assess what goes wrong in the brain on the pathway is going to be probably on the genetics. We're not going to see that on an amyloid or a PET scan. You will have to guess it based on the genetics what goes wrong. So you would treat a diabetic with dyslipidemia differently than a hypertensive person only. So that we have to start doing, and this is what we have to do with the big data.
So just a few short comments. Certainly, as we increase our sample size, the noise does increase, the quality does go down, and yet sample size, as long as it's not too messy, trumps quality in when you get, if you have enough sample size. It's a trade-off, and you have to pay attention to it. But on the other hand, as your sample grows, you can tolerate more noise. Another comment about the pathways that we seem to commonly come up with. There are two comments. First of all, a lot of that pathway identification is pretty soft. Okay, when we talk about lipids, is ApoE a lipid transport protein in Alzheimer's, or is it a scavenger for A beta? Okay, if it's a scavenger for A beta, that changes what pathway it plugs into. Also, much to my disappointment, microglial cells are not just on, off, turn it on, gobble up, turn it off, stop gobbling. They're very complex, and so I think the recent data says, yes, they're part of the pathogenesis, not just the endpoint of the disease process, but we don't understand what to turn on and off in a microglial cell that will benefit Alzheimer's disease. And I think the more detail we can get in terms of what genes are involved, the better chance we have of understanding that.
I'll just very quickly bite on John's note and say that I agree. Perhaps studying these rare phenotypes actually might be more informative than increasing our sample sizes for these more common diseases. I agree with that, yeah.
Okay. Julie.
Several of the speakers have already sort of covered the points that I was going to make, but I think the more power you get in your data set, the more precision you get, and the better quality. So the noise diminishes in its effect sizes. So there is an argument for having large samples because you get more precise information. And I don't think we still know enough about Alzheimer's disease yet, even with the sample sizes that we have. You know, we have 20 or 30 loci that are significant. We know underneath there are others that look very interesting. But which ones do we spend our time looking at in terms of mechanism? These are very expensive studies. Enlarging that population would help that precision. So I don't think we're anywhere near where we could be. And I don't think it's an either or. I think we should do both. We certainly need Lewy body, GWAS, and other rare types of Alzheimer's, early onset, for example. You know, we can learn a lot about what we don't know about early onset Alzheimer's disease, which is considerable. It's very heritable. So there's still a lot to be done. So I don't think we should shoot ourselves in the foot and say we've done enough, because we haven't. There are still big questions to ask there. The other questions that come to mind are rate of decline, psychotic symptoms, depression, irritability, all of those elements we need to get a handle on genetically, and we haven't got big enough samples to look at those.
Take one more question on common variants, and then, yep.
Екатерина Rogaev, University of Toronto, trying to work on my Russian accent here. So it's a question to panel. What do you think about the huge discrepancy between case control studies associated with gene candidate approach versus GWAS finding? The shocking statistic is that only 1% of the gene candidate genes confirmed by large GWAS studies. And is it correct to say that we all were wrong in 99% of the studies previously, or maybe somebody dedicated could look at the past gene candidate studies and say, well, maybe some of the genes are real, but found in a unique ethnic groups, such as Wadi Ara or Arabic population, and to do specific GWAS studies on this population to put the order for the literature to say, yes, it is true, but in a specific subgroup. What do you think?
Well, I think that's a very valid question. I would say that it would be difficult to find the numbers in the specific populations to be able to say a yes or no to each one of those. But of course, that's entirely likely for at least a subset of those previous findings. I would agree.
Yeah, we had a case. It's a pity that, well, we're happy to have José here, but it's a pity that Kari isn't here, because we had a finding in the CHARGE consortium on dementia only in Iceland, and we've been looking for this mutation outside Iceland, and we can't find the mutations anywhere. Also not in Norway, not in Finland, and we only see that in that population, and it makes you wonder what is really happening, right? And I think that John would say you would have to find it somewhere. On the other hand, if we would be so strict on him when if we had been so strict on him when he found APP, he wouldn't have gotten anywhere also. So that is a little bit of a bummer, of course. We weren't then. But with DMD-free, it's really something that we were confronted with, and I think it exists. We're actually we happen to work on a problem in ADSP together with Jerry in the Dutch isolate, and we also see a variant in the location, and we're sure that it must be a mutation there, and we cannot find any other patient series to replicate those, although we see something segregate in the family, and even very strict geneticists from Seattle confirm that it's true, right? So it's a problem, but it also brings us back to the heterogeneity, because if you really look in more in detail, what you think that there should be at least some other variants if it's an ordinary form of disease. But more and more you also start seeing that, of course, in families, the disease doesn't adhere to an ICD classification of disease, right? So a mutation doesn't necessarily present as a perfect Alzheimer or a perfect frontal lobe patient. It just is not the case. And that brings us back again to big data and messy data. I think we need the messiness, the genomics and the biology is going to be messy. It's not going to be strict to certain disease classification as we see them. So in these families, you see dementia and mutations segregate. We come back to that later, but you may see that also in certain populations. But you may not see it elsewhere, and that will be something where the big data setting will help. And what you need to see, if you think it's stratified only in your population as strep stratification, you would expect that somewhere in the world there may be some form of a variant in that region that also gives you some similar phenotype. So, for instance, we see in our family, we see Alzheimer, but we see an association elsewhere with a cognitive function which is really verbally driven. So you must see something somewhere with something else, otherwise it becomes also the likelihood of a false positive becomes very high.
So I'm going to follow up with a question about rare variants, and John.
Actually, I was going to poke you, Kyle, actually.
Okay.
Isn't the problem here the publication process? That everybody tries to get into Nature Genetics when we shouldn't bother with that? We should put the data on bioarchive and then let's see if somebody else does replicate it and not bother with the journals?
That's for me. Okay. Well, I mean, I do agree that bioarchive is changing the publication landscape, and but ultimately every journal has to decide for themselves what their standards are. And the good thing for the authors is that there certainly are a lot of options out there for authors to find, the venue that they think best highlights their work. And so I guess it's a symbiotic relationship that journals have, I think, with the community. And but certainly, as a journal, we're constantly reassessing, publication standards. But I think that from our point of view, the danger of false positive reports getting entrenched, and then being hard to, pull back means that we would rather err on the side of being conservative and, look for well-replicated, robust findings to minimize that, the damage to the community of, thing.
We as a community are too polite.
Shut up, John.
Yes, especially you.
I think you should declare things as wrong, as our opinions about things being wrong. Alex Forum allows you to do that. They edit out a few of the words sometimes, but I think that they do give you the opportunity, which we should use more often, to say, I don't believe this.
I think if I can say something, because I agree with Kyle, Jerry and I lived through the candidate gene period, and you too, John. That was a horrible period, that everybody publishing a gene, and you would think, I pray that not within a week somebody has something out there saying it. I don't see it, right? So I don't think we should go back to this period. It was a horrible time. I think this was the worst decade. The family analysis was fun, the linkage was fun, and then there was this horrible decade, and then we were happy with GWAS again. But I think it will be very confusing if we go back and we have then the duality of the facts and what is knowledge and what is just the fake news out there, right? And that's going to be very difficult also to sell as a public. So I would agree that something of a standard to the community, but also to the clinical community, that you at least to the clinicians say this is the truth, this may be true, and this is something we don't understand. That will be very important because it will be very confusing, I think.
So let me follow up with a question then about rare variants, where of course the challenges of replication become more acute. So with a few notable exceptions, rare variant work in dementia has not been very successful. Do you think large-scale population sequencing studies for dementia will be productive in going after rare variants? Or should we be investing more effort in family-based sequencing studies?
You want to go first?
Yeah. So family-based studies, I don't think we should stop doing them. I think, as Cornelia just mentioned, this is perhaps the most informative approach we have when we have informative families. Large-scale sequencing is something that I'm not sure we're at the point where we should be doing right now. I don't think we've exploited the imputation data sets that we have available as much as we can. And so going after large data sets by whole genome sequencing, I don't think is cost efficient at the moment. That's not to say that I don't think we should do it eventually. I certainly think we should whole genome sequence everyone. Perhaps not with the current technology. If we do that, I'm afraid that in 5 years or so, some company that I will mention will come up with a new method of doing better genomes, and we'll just have to do them all over again. And so until we have a technology that allows us to really, and I'm thinking of phasing data at the moment, you know, we can get short reads and get some information. I guess that 5 years from now, we're going to get different technology that's going to allow us to phase entire chromosomes. This is going to be transformative again. And so at the moment, I don't think that large-scale population sequencing is something, in dementia, is something that we should be doing. I think we should be exploiting more the imputation panels that already exist.
Well, I wouldn't say rare variant studies have been unsuccessful. They've just been underpowered. So we had the IGAP group had a paper that started with exome chip data and ended up with imputed GWAS data all put together. And first of all, the rare variants that we're able to detect, and the ones that have been detected they're not the ApoEs and MAPT of the PSP type odds ratios. They're bigger odds ratios than you get out of GWAS, you know, 1.5 to 2.5, but they're not giant and they're rare. And so in our paper that used really exome chip, de novo sequencing, and then imputed data, we had 70,000 people and I think 30,000 cases, 40,000 controls, and we were able to get significant data on genes that had an odd ratio of 1.5 to 2, but with a low allele frequency of 0.5 to 1% MAF. Not incredibly rare, but that we were at the limits of our sample size of what we were able to detect. So when we say we're having trouble detecting rare variants, I think we've got to increase our sample size. And I think our latest analysis says that in the loci that are sub-threshold, but if you look at them as an aggregate, there's a lot of rare variants that contribute to that, which tells us that the rare variant landscape is definitely part of Alzheimer's disease, and we need a bigger sample size to be able to detect that. And well, I think we're going to talk about imputation later on in terms of how that contributes and how that affects how we do those studies. But basically, rare variant studies are underpowered right now. And I agree with Jose about the family studies. We shouldn't stop them, or else Cornelia would kill me. But we're not going to find another ApoE. We're not going to find a large effect locus. We would have found that by now. We're going to find some interesting biology. And I think all of this, you know, why should we do a larger GWAS study? Will it help us predict Alzheimer's? Probably, as the odds ratios get lower, probably that contribution will be lower. But on the other hand, the effect size doesn't tell you anything about biological significance. So in the cardiovascular world, there was a GWAS of 100,000 people, and 4 of those signals are gene targets in cardiovascular disease. We're not anywhere close to that level of sample size, and also just because it has a small effect size doesn't mean that it's not going to be important in terms of our understanding of the pathogenesis.
I agree with Jerry, of course, and to Jose. No. I think it's a no-brainer that we need to sequence at least more patients. Because the point that Jerry made, and that we made again and again and again, is that, of course, if you're looking for rare variants that cause Alzheimer's disease, the answer will be in the patients with Alzheimer's disease and not so much in sequencing population-based studies. So that is what our money should go, and that is also very disease-specific, I think. So it means that we need to target our diseases and start sequencing large series of patients. For that. The sad story is, of course, that if you think about rare variants, you see mistakes are easier made in rare variant research, both in population-based studies, but also in family-based studies, right? There was this mishap of PLD3, which was partly a family-based study, but couldn't be replicated, not in the population-based studies, but it clearly showed, and we and different groups in Europe and the US showed that it seemed unlikely that this is a causal variant, although it looked at face value as a very validly careful study. And so that was a family-based study, but also in the population-based study, there's mishaps in emerging. What I think is interesting in Alzheimer now is that both the rare variants and the common variants go to the same mechanisms, and that is going to be, again, an interesting phenomenon, and that makes it also interesting to start looking at the pathways more in detail in that sense. And what we should, in the end of the day, try to do, if you think about how they found a cure for dyslipidemia, looking at finding statins, is to get this pathway enhanced and look at the key regulator for that pathway. For coenzyme A, it was only really one target they had to do to block that whole pathway. And that will be the, maybe $100 billion question or pound question. How on earth are we going to find that key regulator? And I think sequencing has a better opportunity to find that than GWAS.
So I'll make a comment and then open it up for more questions. It is interesting to me that although the title of this session is about the feasibility of large-scale genetic risk profiling, it does seem that in the responses from our panel that the reason for doing GWAS or rare variant studies, in your minds, it may be more productive to be thinking about genetics as a way of understanding biology as a path towards intervention and therapeutics, as opposed to genetic discovery for the purposes of being able to do risk profiling in the population. Do you want to comment on those two potential reasons for doing genetics risk, in other words, stratifying the population, predicting risk versus genetics for biological insights?
We just have a paper out in Lancet Neurology, so that builds up also on the US finding that if you look at the risk stratification in the population, we really have a lot of minded still from the common variants, but also of the rare variants. Of course, personally, your rare variant may be a driver, but if you want to know, want to find a high-risk group or low-risk group, for any reason, a low-risk group to understand what protects you from Alzheimer, we probably have that pretty good in our hands. We know that there are people who by age 100 have only a 10% risk of developing Alzheimer's disease, while others, 80% is dead by 80 years old or has the disease. So we have that information at hand, and we can have a lot of mileage by combining ApoE with the common variants there. And having this information in hand is very important to direct effective and powerful preventive trials. And I think if you think about translation, that is probably the way that most of us also in translational sciences want to go, rather to prevention than to cure of the disease, which is going to be difficult.
So I think, risk profiling is important, but it has little meaning if you don't have a therapy to act on. And so chasing after gene targets using genetics, and that's clearly, has a great advantage in terms of success rate in a clinical trial, is having genetic evidence backing your target. Until we have something we can do, risk profile, I don't want to know my risk profile for developing Alzheimer's. I'd rather live in ignorance until there's something I can do about it.
Well, it may be circular, because we never may find an effective drug if we don't know the subgroups. If you would treat a diabetic with an antihypertensive, you probably are not very successful in preventing a myocardial infarction. So we have to match that one way or another with the pathways that go wrong. We all know that it's probably a combination of pathways. It's probably not one pathway, unless you are an amyloid or presenilin-related patient. But you have to have a guess what is wrong in that head, in that brain of that person. And unless we can measure something about the metabolism in the brain that is wrong, either in an exosomes or in a model of a person. The only guess you can have what goes wrong in the metabolism is based on the genes, on the genetic profile. It's a very simple example, but I think I always take this myocardial infarction as an example. So in the old days, you know, grandma will say 20 years ago, if antihypertensives, it was very difficult to show that they protected against against myocardial infarction, because they would have very small effects, but they on an individual still. But if you attack both the fact that somebody has hypertension and has diabetes and has dyslipidemia, then the treatment became effective. And I can't see that Alzheimer's disease is a very much more simple disease, at least in those patients who occur late in life, and that are the most of them.
So, sorry, I'm getting sick of the sound of my own voice. But at what point do you think we can start using, you know, bioinformatics to reduce our Bonferroni score? At what point do you think we can start saying variants in genes we already know might be associated with increased risk? You know, in other words, at what point do you think we can start using other information to try and reduce our number of just raw sequences?
Well, there's been a lot of work done with pathways, and get away from pathways. I had to memorize pathways when I was a student, and that's, you know, it's an old concept. It only rarely applies. It's gene networks. A lot of work has come out of expression profiling, generating these gene networks from the AMP-AD project. And then when you look at those genes, almost none of them come out as genetic signals. And so should we really be using that information? Is it really valuable, trustworthy, reliable stuff we can use? And right now I would think the jury would be out. You know, we all try to use bioinformatics, and as the databases information like GTEx and other like proteomic expression, I think, is a key. As those become richer, they will help us, and as genome-wide confirmation capture becomes available, I think that will help us. And I want to put in one more plug for GWAS, is that, you know, that's the way we get intergenic signals. And everybody says, oh, I want a rare variant. And then you give a rare variant to a biochemist, and I'm a PhD in biochemistry. 10 years later, you say, is that a gain of function or loss of function? Well, I don't know. You know, it's hard. It's hard work. Whereas if you can find an enhancer that upregulates a gene and that's the high risk form, then you know I better downregulate that gene, and you can bypass the agony of biochemistry and then just get straight to trying to design a therapeutic.
I'll just say the danger that is when, you know, to stop doing unbiased studies. You know, if we think this is, it's like just like candidate gene association studies. If we think this is right, we're going to see an effect.
Well, good. I still believe that the genes are a benchmark, so that saved the amyloid hypothesis, I think, and that is why I still believe in amyloid. And I still believe in John, therefore. But I must say that I understand the question also driving it, because I think that the answer should be somewhere in the causomics. That's what everybody feels, has a gut feeling of. But I would say that we have to start working from the genes forward. Then what Jerry just described, that's very difficult. If you start looking at pathways, they are so tissue specific. If you take A's in the brain or A's in the kidney or A's in the vessels, it's going to be very difficult. And we are very naive in biochemistry up until now, what goes wrong in different tissues. So I'm quite sure that the answer to your question is in the fact that we have to do new approaches, probably not so much bioinformatics, but rather big data or deep learning approaches, but starting from the genes and working our way upwards.
So it's been mentioned that imputation can be an effective, cost-effective way to leverage available resources and get the most out of rare variant studies. Can you comment on the current state of imputation in European ancestry populations? Do we have adequate reference panels to accurately impute rare variants? And what about non-European populations?
Do you want?
Sure. Well, we're better than what we were a few years ago. HRC is really, as I think, almost game-changing, with the amount of haplotypes that we have available for European populations, for Caucasian populations. That's not to say that this is enough. We can get down to frequencies that are very low with HRC. Of course, we want to see variants that are even rarer, and so no one is going to say that this is enough. We still need to get better haplotypes, and certainly for other populations where this almost doesn't exist. So I don't think we have reference panels that are adequate for other populations other than Caucasians. Yeah.
I think the other ethnic populations, the TopMed, CCDG consortium, they're working on those panels, and I've seen some unpublished data. I mean, the panels are being formed. They're not going to be as good as the current HRC panel in terms of what you can get down to. And I would say we do need a bigger Caucasian panel in that we want to get down to reliable allele frequencies to match the sample size that we have. So, and the other, even beyond that, if we talk about gene burden analysis, having accurate imputation down to very small allele frequencies will make that possible. Now people are cringe at doing that because of too much inaccurate imputed genotypes. So I think we need bigger Caucasian, and the other ethnicities, we don't have enough sequence data right now to generate the kind of panel we'd really like to have.
No, I agree. I think we worked in another disease, glaucoma, which is actually degeneration of the eye nerve, so that's very related to Alzheimer also. But it's if you go to Africans, where that is a very common disease, it's Africans. You can't do anything with current imputations, and you see really a different pattern emerging. So it will be also to the benefit of the Caucasians to look in this region to sort out also what is the real causal variant. So if anything to invest in Europe, I think there's a lot of ethnic groups that we could, from Pakistan in the UK to Turkish people or Moroccans in the Netherlands or Germany. But I think the Africans scientifically are very important, but they will be quite a job to sequence, because it's going to be a very diverse population, north to south, east to west.
Looking to see if we have any questions on.
Hello, Stefan Ripke here from Berlin. Speaking of the imputation reference, there's always the discussion about like having bigger imputation reference helps you in your imputation. HRC is a perfect example for that. What I'm missing on this conversation a little bit, and I want to ask this to the panel or to others here in the room, is that like imputation quality is not only depending on the imputation reference size, but also on the genotyping platform that the data set is genotyped on. And many data sets that we are still currently working on are like genotyped obviously on older data sets like Affymetrix 500K. Where's the decision on do we want to really put more money into bigger imputation references? I personally think if you start with 200,000 well genotyped SNPs, it's probably not, even bigger imputation reference will not help you very much. You want to have like a bigger data set, and then you want to put your money maybe into these data sets as well. So maybe a discussion point here as well.
Well, there's two issues, of course, right? So if you're interested in disease mechanisms, you want to know that there has been enough Alzheimer patients in the imputation set to pick up that haplotype with the variant, right? So that is something that why I think the imputation sets, you want to impute, if we want to find Alzheimer variants, you want to impute those variants in these sets. So that's the point why I think the bigger the better, and the more patients in there, the better. But I agree with you that some of the arrays are not perfect to do the detailed imputations, right? Whether there is a technical limitation for that, I think there are sufficiently densely genotyped arrays around, right? Whether you take the 1 million or 2 million, but 1 million should be sufficient, I think, for at least Europeans, right? But on the other hand, what I also believe in is like the GSA consortium, that you have a combination of an array with sufficient depth, whether it's 600K, I think it's even also mostly sufficient for Europeans, and some rare variants on there that we know that are key, right? And that are helpful. So I agree with you to some extent, although I think technically there isn't a problem that much. It's more a question on how we spend the money and how expensive these arrays should be.
Well, so I definitely would not disagree at all with if you use the modern chips that are here out there. But I mean, many of these data sets that we're still working on are on these older genotyping chips.
Yeah.
I mean, in my field, like, I totally agree, GSA is perfect, but what's the percentage of GSA right now of these GWAS data sets that I'm working personally on the psychiatric world with lots of data, obviously, and I would say like maybe half of our data sets has like a sufficient modern genotyping platform,
Yeah.
and many others are really old, and I tend to disagree that our great new imputation references would work with these older genotyping platforms.
Well, I have my doubts too, but I must say in my team we are updating the very old array, which we had very old array on. I think we used one from Affim with 50K. We are updating them now with the GSA. So we are doing that now consecutively. Definitely, and I think other people will do that too. But I don't think it's a technical issue, and it's also not so much a money issue. But I think you should need at least 300K, 500K in Europeans in order to do these imputations.
You know, our attitude now is for some of the older data that, you know, GWAS chips are so cheap. If we can get the DNA, which also was done 10, 15 years ago, or I don't know, 10 years ago, it's cost effective to regenotype those same samples, but actually, you know, recollecting those samples into one place is a non-trivial, and we won't be able to do it for all. So, and also for the imputation panels themselves, my understanding the HRC, there's a lot of old data, which is 5x sequence data, not the quality that you would like. So it's a little less than you really think for 30,000, you know, the number 30,000. So I think as the panel gets larger with better quality data, then the imputation will improve. But that also gets into the great heterogeneity of sequence data off of multiple platforms, multiple locations. It's something we're struggling to deal with, but it's a big issue now. And you know, that evil company Illumina keeps coming up with new technologies and new and better and faster technologies, which then are not exactly the same as the old ones, and then we're left with this stew of data that we have to make sense of, and that influences the quality of the reference panel. I mean, more data is all good, it's just difficult to deal with.
Just a counterpoint to that, which is the GSA is really an inexpensive array, and I agree with what you said, older data, crap in, crap out. The GSA is such an inexpensive array that, as Cornelia said, this almost today shouldn't be an issue. If we have old samples and we still have the DNAs, I mean, we should just genotype them on these new arrays. The GSA is one example. For neurodegenerative diseases, there is a narrow X. It's what we're using on all of our samples, simply because, it's 500,000 SNPs, costs us less than 30 quid, and it's worth doing.
Yeah. So, Mike Everly at Illumina. What about, you can create better reference panels by sequencing more, but also what about adding more variant types, which I think Jerry kind of brought up earlier, structural variants. How much improvement could we get just by getting better reference panels, imputation panels for indels of various sizes and different variant types?
Yeah, we want them, right?
Yeah.
It's not so easy. I think we're often stuck in these regions, but I think there has been an effort doing this, I think, also for the imputation panels for the HRC. It's still on the listing, or it should come out still. But it's very important that we start doing that, although it's not an easy task and it's a daunting task to do. But it will be a solution to many problems. But if you have these flagging SNPs and look at these effects of these variants, up until now, what I recall from the statistical analysis, often still the tagging SNP did best, right? So I think in the end of the day, to prove whether it's the tagging SNP or the indel, probably needs some better look either by going back to the lab and reanalyze it, see what really the culprit is, the really causal mutation. But I think it's definitely worth the effort still.
Yeah, if structural variants could be part of the imputation panels, that would be huge. It's been so difficult to reliably call structural variants, even surprisingly the small ones, which you would think would be within a sequence read. You think we should be able to do that, and I don't understand why. It's not a criticism. I just don't understand why we're not very good at doing that. But with the sequence data in-house, existing as methods, I think we'll get better of not only detecting structural variants, but then going back and look, where did that structural variant land in the genome, and what does that area of the genome look like, and is it messy, and is it repeated, and how reliable is this? We will get better. It's just, it takes something different than we're doing right now. But getting that in, but having the SNP array and having the sequence data there, we should be able to go back and put the structural variants into those panels.
Another question? So then maybe also just a comment also, like a technical question or a comment here. So from the Nordic countries, from the Scandinavian countries, they are currently really producing large amounts of sequencing data in there. But they have more like a structural problem in here. So this is data that will not be able to be shipped outside of the country. And so you won't be able to put them all into one place as the HRC did, for example. The HRC did a very smart way of not sharing the original sequence data with everybody who wants to impute, obviously, but they still need technically all the imputation referenced in one place. And this is something that needs to be solved relatively soon. I don't think there is a clear solution to that right now because, again, so these Scandinavian countries, they cannot even share amongst themselves. That's a discussion point there right now. They want to produce, for example, they want to create a Nordic reference data and set, and they're struggling with this because, just by law, they cannot have the sequence data outside of the countries. It's a little bit complicated.
Well, I think we'll discuss this in more detail in the afternoon, the data sharing aspect. But I completely agree. I'll just say that I completely agree that when you think of these large projects, you should certainly, one of the first things you think about should certainly be how you will share data with other people, because there's really, as you just mentioned, this is a perfect example, there's really no sense in doing this, spending all this money and then have it siloed.
In processing sequencing data also, that, you know, there's the problem of when you make a VCF, if you didn't see that variant when you were making that VCF, you're blind as to whether there was good data or no data. And so that's a denominator question. How big is the denominator when you see 0 or 1 copy of that variant? And right now, the only solution is to have all the genomes in one place and called by either recalling them or doing a GVCF type approach where you can go back and get that data for every new variant from every new data set you put in. If you can't bring other data sets together, you will suffer from that. And that's kind of a reality of calling sequence data. And so the Scandinavian genomes, we won't know what the denominator is for some rare variant if they don't have it in their, even if they, well, if they don't have it in their data set.
So, actually, so we'll take one more question.
Yeah.
Yep.
So, hi everybody, I'm Karri Kaivola from the University of Helsinki. I'm here on behalf of Professor Bent Tienri. I would just like to comment on the previous question about the usability of old genotyping chips. Based on our own research we did a couple of months back, we had roughly 300 Finns who had been genotyped using roughly 250,000 SNPs almost 10 years or so ago. And then we had the possibility to whole genome sequence the same 300 people. And then we had the same question in mind, that what are the results if we do imputation based on only the 250,000 SNPs and compare that to the whole genome sequencing? And of course, Finnish population is very homogeneous, and we had a good reference panel of 600 people, but the result was that based only on those 250,000 SNPs, we could impute correctly more than 99% of the genotypes we saw in the BGS. So sometimes even the older genotyping chips really work amazingly well in imputation.
Yeah. Well, you expect that, right? Because the Finns are having big haplotypes shared also because there's so few. But this wouldn't work in Africans, right? So it's also depending on the population.
Yeah, and of course, comment on that, you said that there's a need for a bigger Caucasian reference panel, and I think it would be also noteworthy to build smaller reference panels too, based on the local genomic architecture and the local population, not just creating a bigger and bigger imputation reference panel.
So I'm going to ask one last question to the panel, and this will be on the topic of biobanks, and we'll use UK Biobank as an example. So what are your thoughts on UK Biobank, and how could it be better designed for dementia phenotypes? And maybe the related question is, should we consider pursuing a dementia-focused biobank, and if so, how?
You're UK. Jose. You're not, huh?
I'm not.
So I think UK Biobank has been transformative. I think it's been a new way of doing things, and I think it's incredibly powerful. We were just saying that we need to have patients when we want these, if we want to understand Alzheimer's disease, we need to have patients in these panels. And so I think it kind of makes sense to have the same type of data set for dementia. But at the same time, I think there's a lot we can take out of the UK Biobank before we go into something like this.
Yeah, I think there's also the dementia platform, right, of the MRC that of Gallagher so that there is another. I think it's definitely interesting for any population to have a larger catchment area for patients and to connect that also with biobanking. I think in the end of the day, most populations will be at least erased, almost completely. So it would be then a no-brainer to make these connections also to the patients. But it's definitely not an easy task. If I look at the dementia platform, it's not so easy to catch the patient. I see Julie nodding. I know you're in it. Because I think what you will find is also a lot of oddities in there, and that is probably the same thing as we found in an epidemiological population-based settings. You're probably not going to find the pure Alzheimer's that you find in your clinics. And that is something that also makes it more interesting, it makes it more noisy, it makes it more big data, but it definitely brings us back to the sobering fact of life. That was always brought up by John Ioannidis, who said, well, if you look at the population, it's not so pure as you would like it to be. So also in the population, you will see a lot of different phenotypes, because I think you guys see now a lot of Alzheimer, alcohol-related dementias, right?
So one of the advantages both DPU UK and Biobank are epidemiologically based. So this is a very different sort of question. I think if you were designing something for genetics, you would want to have the ability to go back to people on the basis of their genotype. So we want to go back and test people for certain clinical features that the mechanisms that we're working on seem to indicate, for example. So I think that would be one very important element of anything that is genetically based. It's not so easy to do that with epidemiological studies, because you don't want to go in there with a clinical trial, for example. It is challenging to change the epidemiology profile of those cohorts. It doesn't always work. It works better, actually, with people that are not working on dementia as their main focus. Those were set up to try and look at the relationship between genetics and epidemiology, amongst many other things. But from a genetic point of view, that's the use, I think, of both DPUK and Biobank. But if you were going for something for a genetic set of cohorts, then I would go for something a little different.
So it's interesting how much, in looking at the UK Biobank using a phenotype of dementia, how much that agreed with what we find in Alzheimer's disease.
Yes.
And it almost makes you wonder, maybe we're shooting ourselves in the foot by insisting on pure Alzheimer's disease. Maybe what we should be studying is dementia. And maybe that's not the noise. Maybe that's what we should be looking at.
Well,
Maybe the Alzheimer is noisy.
However, I think we need to go both ways. We need to go for greater precision. And what that showed us was that if you have very, very large samples, then you can deal with the noise that you get, because you've got a signal in there that is somewhat similar to our clinical diagnosis of Alzheimer's disease. But it's going to be a lot noisier, and for that reason, you need very large samples. And if you've got very large samples, that's fine. But I think we also need to go for the precision and look for specific features. Early onset Alzheimer's is something I'm interested in, because I think it's highly heritable. If you can get power behind those sorts of samples, you can get greater precision about what is associated in those groups, and that will help with other mechanistic studies and fill in the gaps that we know are there. So I think we need to come at it from all angles. But we only need a smaller sample to do that in relative terms to the very large sample that you would have to collect for a very noisy phenotype, like any sort, or your grandparents or your parents' diagnosis of dementia, which is what that was based on. So it's horses for courses.
When I say something curmudgeonly about UK Biobank and such similar things, of course, it's good to have open access data. What I will say is it's moving the centre of power from those of who have worked on Alzheimer's disease to people who sit at computers and get access to data downloads. And one might think that's okay, but it really is devaluing the generation of data in a sense. And that's something we have to think about. We who generate data have to deal with all the HR issues of hiring, of firing people, and all this sort of stuff. Whereas I just feel that there is an army of people just waiting to download the data without any of these issues hindering them. And, I feel sorry for myself.
And they're younger, smarter, and better looking than us.
It's welcome to the 21st century, John. But it's...
John, just in addition to that, of course that's an issue, but it's also an issue that those people won't have the biological background that people that have been working in Alzheimer's and neurodegenerative diseases have. And so the interpretation of the results and the data may be completely different, I would say, or at least a little bit different.
Well, what I thought is surprising thing, that you have these very small effects that you wouldn't think segregating over families, right? I'm looking at John because he works with families too, Jerry. You wouldn't have expected that, right? That you see these small effects that you can still pick them up with family history. It really is amazing. So it gives us the power, and I told you, John, and Julie said the same, it's the power of the big data. It's the power of the big data, and there must be an answer in it. And I think it's comforting to see that these young people are sitting and waiting and have a big enough computer to do the analysis, because not a lot that can do that. They still come with the same answers. So it means that we're doing something right? So it means that this is the truth.
So it's Valentina Escott-Price. Adding to Rita's point, it's not only people sitting and waiting, downloading, and which are not a specialist in Alzheimer's disease. Sometimes in terms of data analysis, they're setting up standard pipelines and actually treating variables, each variable in the same way, and they do come with wrong conclusions. And in my experience, I'm at the moment, one result was published and I analyzing data my way, well, taking into account all variability. I find opposite result. So it does happen that those people who are just downloading data without thinking and setting standard pipeline analysis, they come with wrong conclusions.
Yeah, and I'll add a comment. Also from a publishing point of view, of course, it becomes a challenging thing when multiple groups simultaneously have access to the same data, and there are certainly ways to analyze the data to generate, a manuscript for consideration. And so it becomes an interesting question of what the journals will use to distinguish one analysis from another. And I can give an example, from a different community, the metabolic disease community, where we've seen, parallel submissions of analyses of UK Biobank data from groups with disease expertise versus secondary users that don't have a metabolic disease background. And it definitely shows in the quality of the biological interpretations and also the access to additional supporting data sets, for instance, islet expression, data, and the ways to actually get more out of the GWAS signals than just reporting where the signals are. So I certainly would advocate on behalf of the disease communities that, and say that as journals, we see the added value that, the disease relevant expertise adds to the quality of the analysis, and certainly want to continue to encourage the disease experts to, Yes, hang in there, guys, hang in there.
But it's interesting, Carl. Can we ask you, because you must have this different groups sending up the same observation, right? Or partly overlapping findings from the same data. So how do you treat that? You just accept them both, or do you say merge them? It's very, because it becomes very confusing to interpret it as a readership. Are these two independent findings, or am I looking at conflicting findings, or is it twice the same?
We're trying to work through that question. I mean, what I can say is we are not in the practice of asking two groups to get together and come back with a new, combined submission, partly because we understand that that's not how these projects evolved, and it's rather unfair at the end point that we see a manuscript to actually tell them, they need to, and it would breach many levels of confidentiality when we receive a manuscript.
Yeah. On the other hand,
so really, I think the best that we can do is handle each submission fairly, judging it on its own merit. If they happen to be submitted to us in a comparable time frame, we will make every effort, if they make it through peer review, to coordinate publication, because we think that's in the best interests of the communities to have them come out together. But yeah, it becomes much more challenging when you get slightly asynchronous submissions, so that they're not on quite the same time frame, and they might be of uneven quality, and then, where do you draw distinctions? But we try and, again, make as fair judgments that we can, taking into consideration all those complexities,
Well, you would think that at 21st century, you have this common shared data set, that you would also have a community sense that even post hoc you should work together to help John with his frustrations, right? That you say, listen, this is open data, and now you have to have an open community effort also. I was just wondering,
So I think we're going to talk about data sharing, but data sharing is not equal across different countries. And, DNA sequence data, we're required by NIH we have, certain number of months after we get the data to allow us to actually call and process the data, and then it has to go out. And for the Alzheimer's disease sequencing project, ADSP, we very strictly adhered to that NIH rule. And so we had the data to analyze pretty much the same time as the rest of the world had the data to analyze. But that's not true of data generated in other countries. It's a very unfair landscape, and I think it inhibits collaboration in terms of, well, we better not collaborate and keep our data and analyze it ourselves as fast as we can, versus putting it out there so it all could be analyzed as one common data set, because we can't get our hands on data sets from certain countries.
Yeah, and so on that point, and especially since we have a session on data sharing this afternoon, we'll be able to definitely pick up that point and kind of explore that more. So I'm going to close this session now, thanking again our panelists for sharing their very thoughtful insights into the state of genetic risk profiling and discovery. I thank all of you in the audience for the very thought-provoking questions.